Hi, this is Professor Tiffany Martin with Medical Microbiology, and we are in Chapter 13 talking about viruses. And we have just finished talking about animal viruses. We've talked about attachment and entry of animal viruses. Now we're going to move on to synthesis. And remember, synthesis is a stage where it needs to make viral proteins and it needs to copy viral nucleic acid. So let's talk about the different types of nucleic acid that comes in and how that is affected there. Okay, so synthesis of animal viruses. So we have got a virus and it has used one of those methods to get inside an animal cell. Now, unlike a prokaryotic cell, a eukaryotic cell is much larger and it's got compartments, right? It's got a nucleus, it's got mitochondria, it's got a lot of other things. So there's a lot of choices that viruses have on where it stays. And what we have observed is that if the viral nucleic acid is DNA, that virus, once it gets inside, will move into the nucleus, the nucleic acid, the DNA moves into the nucleus, and then everything happens in the nucleus. If the viral nucleic acid comes into the host cell and it is RNA viral nucleic acid, it doesn't go into the nucleus, it actually stays in the cytoplasm. All right, so you should know that the type of nucleic acid in the virus depends on where this virus is going to go in a eukaryotic cell and replicate and work. So DNA viruses enter the host cell nucleus and they are replicated and assembled there, which kind of makes sense because that's where the DNA is. That's where the enzymes involved in the host cell DNA replication is. So it makes sense. DNA viruses go into the nucleus. RNA viruses, they stay in the cytoplasm, which that's where the ribosomes are, and that's where RNA is. And so they are replicated and assembled in the cytoplasm. Okay, so let's talk a little bit more about the different types of viruses we have and where they're going to go. So DNA viruses, where are they going to go? They're going to go into the nucleus. What happens in the nucleus? We need to replicate the DNA and undergo transcription, make mRNA in translation. So a double-stranded viral DNA molecule comes into the nucleus. Well, the host cell knows what to do with that. We have double-stranded DNA. Pretty much transcription and translation happens as normal, uses the same enzymes that we just memorized to copy it. Helicase, RNA polymerase for transcription, DNA polymerase 3 for replication. All of those enzymes work and we end up making mRNA, which will then go out into the cytoplasm and make proteins, and we end up replicating DNA. So all of that happens pretty much as normal. Now, if the viral nucleic acid is single-stranded DNA, it's DNA, so where's it going to go? The nucleus, but we don't really have single-stranded DNA as much. We have some, but there's a little bit of things that have to go on. Single-stranded DNA, that will, an RNA polymerase will bind to single-stranded DNA and make mRNA. And then also, it will make the single-stranded DNA kind of fold over on itself, because remember, in DNA replication, we need a primer. The single-stranded piece of DNA kind of folds over itself and then uses that as a primer and makes a template copy that then can be used to make a whole bunch of more single-stranded DNA. Now remember, single-stranded DNA, it makes a difference whether it's the sense strand or the other strand, and so it's got to make sure that it copies the same strand that comes in. The single-stranded DNA that comes in is going to make a complementary copy of itself, and then that complementary copy is going to be used, complementary base pairing, to make a whole bunch of whatever came in. All right, so basically double-stranded DNA and single-stranded DNA go into the nucleus and act as normal. A little bit of maneuvering, but basically the enzymes, the host cell enzymes know what to do with that. So now let's talk about RNA viruses. RNA viruses, they come in, where are they going to work? They're going to stay in the cytoplasm because they're RNA, but RNA can also be designated positive or negative. Remember the sense strand and the non-sense strand? Positive RNA viruses, or we could have the opposite of that and the negative strand RNA viruses. What's the difference? A positive single-stranded RNA can act directly as mRNA. So what do I mean by that? The strand of RNA can go straight, that comes in, the viral nucleic acid can go straight to a ribosome and gets translated into a correct viral protein. We call that a positive single-stranded RNA. It is the RNA that is equivalent to a strand of mRNA, meaning it can go into a ribosome and be directly translated into a correct viral protein. So that's positive single-stranded RNA. So that comes in and that has to be done. Positive single-stranded RNA. Sometimes some virions, some different types of viruses, come in with a negative single-stranded RNA. Negative single-stranded RNA can't bind to a ribosome and get translated. It will make a completely different protein. Remember, it's got the opposite complementary base pairs. So negative single-stranded RNA first has to be copied into a positive single-stranded RNA, and then it can be used as a protein by a ribosome to make a protein. Positive single-stranded RNA can be directly translated. Negative single-stranded RNA has to go through a intermediate, has to be copied using complementary base pairing to make a positive single-stranded RNA, and then that can be used to make proteins. So that takes care of protein synthesis, but we also need nucleic acid synthesis. We need to make a whole bunch of nucleic acids, RNA, to get packaged into these virions or these viruses that are going to be released as virions. So we also need to make a whole bunch of RNA. So if it came in as positive single-stranded RNA, we need to make a whole bunch more positive single-stranded RNA to go out. How do we copy nucleic acid? We got to make a template. We have to make a complementary copy and then use that as a template to make a whole bunch more. So if it comes in as positive single-stranded RNA, it's then going to make a complementary negative single-stranded RNA, and then that is used to make a whole bunch more positive single-stranded RNA. Okay, all right, so opposite, if it comes in as negative single-stranded RNA, we got to create a whole bunch of negative single-stranded RNA to go out. So a negative single-stranded RNA, it's going to make a positive single-stranded RNA to use as a template, and then that can churn out a whole bunch of copies of negative RNA. All right, I think I tried to write this on the whiteboard. We'll see how I did. If we're talking DNA, we have double-stranded DNA, which basically acts as normal. We have single-stranded DNA, which can then be transcribed into mRNA. And then that single-stranded DNA has to loop around and do some things to make more single-stranded DNA to come out. Let's do single-stranded RNA from that. Here, I'll show the whole thing. Oh, too many words on this board. I'm so sorry. All right, let's do single-stranded RNA. Single-stranded RNA can come in as either two types. Sometimes single-stranded RNA for some viruses, we call it positive single-stranded RNA. Some viruses come in with negative single-stranded RNA. What's the difference? Positive single-stranded RNA is equivalent to mRNA, meaning it comes in and it can immediately start making proteins. Negative single-stranded RNA, before anything can happen with a negative single-stranded RNA, it needs to make a complementary copy. Needs to use the enzymes to make a positive single-stranded RNA. Now then, this positive single-stranded RNA can be used directly as mRNA, and either way, we have to use the complement to make what came in. So again, I've got to copy my nucleic acid, and the only way I can make more of this is by going through a template and then churning out a bunch of this. The only way I can make more of this is by going through a template and then churning out a bunch of this. Okay, all right, so going through and copying single-stranded RNA, we've got some different things that need to be understood. All right, bring your questions to that on me, talk about it in study groups, and we'll see if we can make sure we understand that. All right, so going back to my PowerPoints and summary kind of PowerPoints, synthesis of positive RNA. Positive RNA can go directly to a protein or to a ribosome and get translated into a protein, but we still need to create a negative single strand so we can make a whole bunch of positive single strands. We're going to come back to retroviruses, so we'll come back to that in a minute. If I'm doing a negative single-stranded RNA, then I still need to go through a positive single-stranded RNA first, and then that can be used as mRNA, and it can also be used as a template to create more single-stranded RNA. Now, there is also viruses that come in as double-stranded RNA. So this is something that's different. We've not seen, we don't really have double-stranded RNA. Well, I guess some of the tRNAs, but anyway, double-stranded RNA can come in. How does that work? Double-stranded RNA, what we observe happens is once a double-stranded RNA virion comes in, it's RNA, so where's it going to stay? Cytoplasm, right? Not nucleus, it's RNA, so it's going to stay in the cytoplasm. Once it comes inside in the cytoplasm, it actually gets unwound into two strands of RNA. One is positive and one is negative. So that happens. Now the positive can be used directly on a ribosome to create proteins, but we still have to create double-stranded RNA to get out. So we still need to copy both of these. What we see happening, what we observe, is the positive single-stranded RNA gets used to make a negative single-stranded RNA. And then the negative single-stranded RNA gets used to make a positive single-stranded RNA. And then these two come together to make the double-stranded RNA that it was coming in with. So that's what we see for that. It gets copied and makes a whole bunch of positive and negatives, and those positive and negatives, due to complementary base pairing, come together to make a double-stranded RNA. Whew. Okay, don't let me lose you on this. Go back and review what you need. Okay, so let's do some pictures. Let's do some pictures because sometimes pictures are easier. And let's talk about this. Okay, so we have a picture of entry and synthesis going on. So just by the picture, I have viral DNA here drawn in pink. We can tell it's double-stranded because it's drawn that way. We can also tell what about it. If this is the nuclear membrane, there's a nuclear pore, we know that this is double-stranded DNA virus, nucleic acid that's coming in just based on the pictures because it's going into the nucleus and DNA goes into the nucleus to work. What happens when it gets into the nucleus? So it's gotten into the nucleus. Then transcription is going to occur, just like normal. We know what to do with double-stranded DNA. We're going to make mRNA. That's going to go out and get translated into viral proteins. Okay, so that makes sense. What else happens? Well, we need to copy this, so we need DNA replication to occur. And that happens just like regular DNA replication. It's going to unwind and use semi-conservative base pairing to create double-stranded DNA. So a whole bunch of that happens. Now what we also see is these viral proteins that got made in the cytoplasm. They actually get shipped back into the nucleus, not back in, but they get shipped into the nucleus, and we see assembly happening here. So for DNA viruses, everything happens in the nucleus. They go into the nucleus, replication happens there, mRNA gets made. Now, yes, that gets shipped out to the ribosomes, but then the resulting products, the proteins, come back in, and assembly occurs in the nucleus. So we make these mature viruses that are then going to get taken out of the cell. We'll talk about that in a minute. All right, so this is double-stranded DNA. But some virions, or some viruses that have double-stranded DNA, also have the ability to integrate into the host cell DNA. You remember our prophage, when a bacteriophage integrated into the lysogeny? Now, this is a different type of cell, so we're going to give it some different terminology, some different vocabulary. But if the viral nucleic acid, the viral DNA, integrates in the host DNA, we're going to call this provirus. So the viral DNA is now referred to as a provirus, not a prophage, that was bacteriophage, but a provirus. The other thing is this is not called lysogeny. This is also actually called latency. We're going to see some examples of some different viruses that undergo a latent phase, and they work by integrating into our host DNA. So that's something that can happen there. All right, how was that picture? Let's do this one. This one, we have a virus coming in. You immediately notice that it's single-stranded. And also from the picture, you can tell this is an RNA virus because it's happening in the cytoplasm. Now, in this case, this is positive single-stranded RNA. So positive single-stranded RNA can be used as mRNA and get immediately translated into proteins. We also need to make a negative single-stranded RNA template so we can make a whole bunch more positive single-stranded RNA to get assembled into viruses. All right, how is that picture? Let's do one more picture. This is a big one. Stay with me. So positive single-stranded RNA, we've just seen this one. This is a different type of picture. I think this is the one in your textbook. We've got attachment and we have entry and the positive single-stranded RNA. This looks like, ooh, what type of entry method is this? Ooh, I hope you said direct penetration because you can see it's a capsid, not an envelope. It's getting penetrating into the membrane. So this is a direct penetration. It's the nucleic acid that enters. I have positive single-stranded RNA that enters. So what happens? We're going to make a complementary negative single-stranded RNA. So we can make a whole bunch of positive single-stranded RNA. Some of that positive single-stranded RNA can go straight to a ribosome and make proteins, and then we get assembly. So that's a positive single-stranded RNA virus. Negative single-stranded RNA virus. We go into here, negative single-stranded, first thing that has to happen is we need to make a complementary positive single-stranded RNA. Then we can make proteins. Then we can make a whole bunch of negative single-stranded RNA to get assembled and released. Double-stranded RNA virus. This is the one where I told you it unwinds. And then we can use that as mRNA. And then we can make a whole bunch of complementary RNA strands that wind back together to get assembly. Okay. All right. So what did I miss? I told you I'd go back to it, retroviruses. So let's talk for just a second about retroviruses. Retroviruses are actually positive single-stranded RNA viruses. So they come in, but unlike most positive single-stranded RNA viruses, which work in the cytoplasm and they make that negative single-stranded RNA, positive single-stranded RNA retroviruses are actually get used to create inside the cell double-stranded DNA. And they do this by bringing their own enzyme called reverse transcriptase. So again, does a cell, do we ever go from RNA to DNA? No, central dogma, DNA to RNA to protein. We don't have any enzymes. We don't have any tools that are capable of taking an RNA strand and making DNA out of it. So a retrovirus needs to bring its own enzyme with it, gets packaged in the virion, and it makes, or it may make it as positive single-stranded RNA right when it gets inside the cell, and it makes this enzyme called reverse transcriptase. This reverse transcriptase can take this RNA and use it as a template to make double-stranded DNA. Why is this a big deal? DNA viruses, many DNA viruses are capable of this integration. And when it integrates, it hides, and we can't find it. We can't get rid of it very easily. It can stay in here. Remember lysogeny, replication goes on. It can stay in here, and it can come back. And that can be an issue. Retroviruses that are causes problems. HIV is a retrovirus. So any retroviruses can be potentially damaging and very concerning. Any DNA viruses that can integrate can stay with us and cause a long time. All right, let's stop there. That was a big one. So get with your study groups and talk this out and bring me your questions. Okay, good job staying with me. Thanks.